Hello everyone, thank you for joining us for this Precision Proteomics webinar on how to identify biomarkers in health and diseases. My name is Joel. I'm your technical application scientist from Azenta, and I will be your moderator today. Today's webinar will be led by Dr. Michael Förster, who is the Director in Oncology and Rare Disease of the Scientific Affairs team in Olink Proteomics. He completed his PhD on autoimmune disease genetics at Karolinska Institute and moved to McGill University for a postdoctoral position, investigating the link between primary immunodeficiency, hematopoiesis, and the development of hematological malignancies. He left academia for a stint at the Fraunhofer Society to manage projects in liquid biopsy-based cancer diagnostics and CAR T-cell therapy, and more recently worked for Azenta Life Science as scientific liaison for biospecimen procurement and genomics services. Now, throughout the event, please feel free to send any questions you may have through the question chat box. Dr. Förster and I will address your questions during the Q&A portion of the webinar. Without further ado, I would like to hand everything over to Dr. Förster to begin the presentation.
Yeah, thanks so much, Joe, for the kind introduction. As you already mentioned, my name is Michael Förster. I'm Scientific Director for Oncology and Rare Disease at Olink Proteomics, and I'll talk to you today about precision proteomics and how that enables biomarker identification in both like health and disease. So, starting out, I actually would like to quote developmental biologist extraordinaire and Nobel Prize laureate Sidney Brenner, who said that progress in science depends on new techniques and new discoveries and new ideas, probably in that order. And I hope by the end of that webinar, I will have convinced you that Olink and the Proximity Extension Assay that is the basis for our products is one such new technology that will enable you to hopefully make new discoveries. So without further ado, like what is our company's vision? I mean, we really hope to enable the understanding of real-time human biology by using proteomics. And we do think that the multi-omics approach, combining different omics technologies, holds a lot of promise, but going into like the way back machine of genomics and epigenomics and the way back machine of transcriptomics, we think that proteomics as an additional layer on top actually will help to complement and fill this, I mean, complete this picture of human biology, and then what goes wrong, not only, I mean, like in disease conditions and enable a better understanding of disease, fundamentally also informing, hopefully, a better treatment for patients. So where do we do what we do? By trade, I mean, like originally we're a Swedish company. We've been founded in Uppsala, basically coming out of Ulf Landegren's laboratory at the Uppsala SciLife Lab. So he developed the original proximity ligation assay and the proximity extension assay. And we do have worldwide presence. What is most important in the context of this webinar is that Azenta has brought in the technology and offers the proximity extension assay product line from their laboratories in New Jersey, so you can basically send your samples to them, have them analyzed, get the data back on a relatively simple fee-for-service arrangement. So what is the proximity extension assay? So the proximity extension assay, I hope you can see my video here, is basically a dual antibody capture proteomics assay, meaning that two antibodies have to bind a protein analyte at the same time. Those antibodies, as you can see, are DNA tagged that will facilitate complementary base pair binding. You will then have a ligation reaction. That ligation reaction will generate double-stranded DNA barcodes, and these DNA barcodes, they contain these small adapter molecules that are depicted just in a second here, and these ones are used as targets for the sequencing reaction, either and then the readout either via NGS or via qPCR. So this principle for the proximity extension assay has, as we see it, 5 big advantages. The first advantage is clearly specificity, because you have to have 2 antibodies that bind the same protein analyte. You're basically guaranteed that you really measure what you want to measure. Then secondly is you have increased sensitivity. That's just the nature of the NGS or the qPCR readout, and the exponential de novo synthesis of nucleic acid that helps us with the sensitivity. And since you basically, I mean, in theory, you don't really are kind of like hampered or by the availability of either antibodies or DNA barcodes, you can actually multiplex in the reaction, so you can measure multiple proteins in the same reaction, and then at the same time also do that in a very reduced amount of volume. So for us, 1 to 1.5 microliters of cellular plasma per reaction is totally enough to read out at least 384 proteins in one reaction. And then, since it is an immunoassay, that is the underlying principle, we do this. I mean, there is a possibility to implement it throughout the entire value chain, and kind of like easily implemented into the clinic as well, because the FDA regulations, or the regulations for immunoassays, are very well known, and our validation process actually follows those as well. So what is the product? So what can you read out at the moment? Here's the product portfolio. It's kind of neatly bifurcated by readout. So on the left side, what can you do with the NGS readout? So that's our discovery platform. It's called Olink Explore. Right now, you can read out up to 3,000 proteins. Not all 3,000 proteins in a single reaction. You do that blockwise. So one reaction is 384 proteins. We have 8 blocks that adds up to 3,000 proteins. We have 2 oncology blocks, 2 immunology inflammation blocks, 2 neurology blocks with neuronal genes, and 1 with cardiovascular and metabolism proteins. Mainly. So you don't really have to run the entire discovery platform all the time. There is some modularity involved as well, which should help with costs if that's needed. And then on the right side, you see the qPCR readout, and here we have the Target 48 platform. So the Target 48 platform is our 48-plex cytokine panel, mid-plex panel, where you can read out cytokines either in relative units as NPX or with absolute quantification. And then we do have 15 Target 96 panels. So these ones are pre-spotted panels where you can read out immuno-oncology targets. You can read out inflammation targets, you can read out neurology targets, cardiovascular disease, cell regulation, development, different types of plates, a bit more directed assays. And then on the extreme right, there's the focus panel. So that's kind of like our stab at towards clinical translation. So let's assume you do a discovery experiment, you find a biomarker signature, you want to measure thousands of patients to validate it. The focus panel allows you to do that by combining 21 proteins and either read those in absolute or in relative units. So you do have two validation levels. This is a product that's going to be released by the end of the year. So that's going to be available very soon. And for the Explore panel, I can assure you that we are working very hard to extend the library. So actually by the end of the year, it's not only going to be 3,000 proteins, it's actually going to be 4,500 proteins. So there are going to be 1,500 additional targets. And the qPCR assays you can neatly read out on the Signature Q100. So that's our own in-house development. It's a qPCR machine. We developed that together with Fluidigm / what is now Standard BioTools, and Azenta has those available, so they can offer the qPCR product line, and they will very soon be able to offer the NGS product line as well. So from that, question you may ask is what's included in the library. So what's currently included is 3,000 proteins. Of those, approximately 1,400 are new biomarkers. We do develop our own antibodies, so 900 of the antibody pairs are proprietary Olink antibodies. We do have customer wish markers, so we do add, with feedback from scientists, specific proteins to library extensions as well. We have done that in the last 2 iterations. We aim to do that for the next iteration as well. And you can actually check the entire library for your favorite protein, and should it not be included, you can reach out either to technical service or to scientific affairs at Olink, and we will do our best to accommodate you in future libraries if there is an an economic strong interest for this protein. We also have 60 high abundant proteins. So those ones, why would you add those? Well, those ones are basically proteins that you can easily pick up by mass spectrometry, so by more established proteomic technologies, you do have a possibility to have orthogonal validation and basically validate targets that you should be able to pick up with any mass spec experiment with Olink, which we kind of do to increase the confidence in the data that you can derive with our library. In itself, we have 450 proteins that are associated with the human secretome. We have 250 organ-specific blood biomarkers that you can pick up in blood. We have a broad range of inflammatory proteins, so up to 1,200 of our targets are actually in one way or the other related to inflammation. And we do have 600 established drug targets as well. And then, 800 proteins that are in one way or the other in exploratory ways have been annotated for blood and in circulation. So then on the topic of orthogonal comparisons, so what people have done, especially for our cytokine assay, that was the first product that Olink launched, they have compared us to both Luminix and MSD. These are independent comparisons. This is not experiments that Olink has undertaken. This is independent data derived from other labs that had those technologies available. And we had better sensitivity, especially for low abundant cytokines. We had a wider dynamic range. We had best linearity and in general highest quality when it came to cytokine readouts. So if you currently do a lot of cytokine measurements, for example, in preclinical models, you could do that with Olink as well, and you could use us for that in the target 48 iteration. And then the other orthogonal comparison that we are often asked for is mass spec. So in terms of how do we compare to mass spectrometry data, so with mass spectrometry, kind of like what you will find is you can pick up anywhere between like 300 to 600 proteins in circulation. Most of those will be classical plasma proteins like LDL or HDL, and there will be a couple of tissue leakage proteins, like for example, PSA or troponin, things like that. So that's what you can pick up with mass spectrometry, and that you will also be able to pick up with Olink's library, because a lot of those targets are included in our library as well. However, we do complement mass spec data just because we have a broader library and we do have a broader dynamic range, so you can really extend your analysis of the plasma proteome to low abundant proteins, for example, proteins that you wouldn't be able to pick up with a mass spec experiment, like low abundant cytokines like IL-8, or things like neurofilament light chain in circulation that is somewhere in the femtogram range. So that's how we can help proteomics researchers that are doing a lot of mass spec experiments to basically extend their breadth of analysis. And then in terms of sample types, so the library, as I already mentioned, is geared towards like the plasma or serum proteome in circulation. That's the initial validation step we always do for any new target. Is it, can you detect it in different forms of plasma or in serum? All neurology targets are additionally validated with cerebrospinal fluid, so CSF. But that does not mean that you cannot use our assays for other bodily fluids or other like biospecimen types. So we have been used in broader contexts for 20 plus different matrices, anything from urine to saliva to tear fluid, dried blood spots, bulk, blister fluid, CSF, and also cell and tissue lysates and cell culture media. There are only two kind of like biospecimen that we do not recommend you use the assays for. And that is anything that's formalin fixed. So formalin fixed tissue, even if you could measure it, the formalin fixation actually destroys too many of the antibody epitopes. So that's does not lend itself for the Olink assays. And then the other thing that people have used us for is fecal matter, which unfortunately contains too much complex polysaccharides that unfortunately interfere with antibody binding as well. And then you will not have a stable and good readout for the proximity extension assay. So we do not recommend that. We do not recommend that either. So this is just an overview slide in terms of we are at present stage, we have analyzed more than 3 million samples in our own service labs, and many more in our partner labs, including Azenta. And at present stage, Olink has been used in more than 900 publications. I mean, the read a couple of weeks ago was something around like 925. We do have our own consortia that I will mention a little bit later, and we offer a broad range of complete technical validation. So we help you if you're thinking about doing experiments on a new biospecimen type. So we help you with that. And we, of course, also validate any of the new antibodies for library extension. So that's pretty much the product line of Olink. And now I've come more to the scientific part of my talk in terms of how do we support the drug development process. We do support the entire value chain from discovery to kind of like FDA approval, like post-market surveillance steps. And of course, one has to be realistic. We have been used a lot in the discovery stage, so people want to use us for identification of new drug targets. And they also use us a lot for identifying biomarker signature in a clinical context. So even in clinical trials, I mean, the current number, currently there are 37 clinical trials that have incorporated Olink assays to stratify patients in one way or the other. So that's kind of like, despite us being just a research use only application. We have also been used in preclinical research, so in vitro and in vivo testing, looking for biomarkers, even in xenotransplantation models. We do have one mouse panel, one target 96 panel that's specifically for mouse proteins available, and we also validated the library for non-human primates. So should you be interested in experiments with non-human primates, be it in the context of, for example, infectious diseases, Olink also has successfully been used within that. And I will, in the second part of my talk, give you an example or a couple of examples for all of these parts of the value chain. So starting out with proteogenomics, the signature project for us is the Pharma Proteomics project. The Pharma Proteomics project is basically the UK biobank, the biggest biobank in the world, with support from more than a dozen pharma companies, made us their preferred proteomics partner. And what they did is they analyzed approximately 1/10 of their biobank, so more than 53,000 individuals with the Explore 1.5K iteration of the discovery assay. So that's kind of what has been done. And 2 weeks ago, the initial manuscript was actually published. So it's currently under review and was actually published on Bioarchive. So here, Benjamin Sun and colleagues report the genetic regulation of the plasma proteome for this UK biobank cohort. And in terms of what they find, I mean, first the study design. So they chose approximately 54,000 individuals. They did analyze 1,463 proteins. The mean coefficient of variation, so kind of like the intra-sample variation, so technical replicates for duplicate samples for the same target spread over different plates, was 6.2%, which is excellent and actually comparable with top industry standard. And the genotyping was approximately 22.2 million imputed SNPs. So of the biobank cohort, what they did is they split it in 3 different subcohorts. So first, they did a populational assessment where they just randomly selected samples. It was 46,600 samples. And then they had the diagnostics and disease part of the cohort. So that was approximately 6,400 individuals that were enriched for 122 diseases. They were a little bit different from the general population. In the cohort, they had a higher BMI, they had a lower proportion of females. They were a little bit older, higher smoke prevalence, and they were a little bit more multi-ethnic when assessing ethnicity by self-report. And then lastly, they actually also added like the biggest COVID cohort. So they had COVID-19 patients, approximately 1,300 coming from a serial MRI, a serial imaging study in the UK. They were also a little bit different from the general population in that their BMI was lower. They were much younger by 6.3 years, and they had a lower smoking prevalence. But they were all COVID patients, and of course, they had medical outcome for COVID-19 associated. What they wanted to do is they wanted to find meaningful genotype, protein, phenotype triads. So basically genomic signals that control protein expression in circulation and hopefully overlap with complex phenotypes. And for the bioinformatic analysis, they split their entire cohort into a discovery part. That discovery cohort was basically their white Englander population, and then their validation cohort was everybody else who was a little bit more multi-ethnic. So I would like to you to keep in mind that these two cohorts, the split that were the validation cohort is a little bit more genetically diverse than the discovery cohort. So what did they find? Of course, they had it's a very, very long and complicated paper, but one of the things that this paper shows is that's really the advent of systems biology and proteogenomics. So they basically looked at circulating proteins like cytokines or complement. I show you in the cytokine example. And what they found is here shown for IL-15, they found cis-pQTLs that basically regulate IL-15 at the IL-15 locus, but they also found 4 trans-pQTLs in the IL-15 signaling pathway. So they basically found SNPs, both rare and common, some intronic, some coding, in the IL-15 receptor alpha chain, the IL-15 receptor beta chain that's shared with the IL-2 receptor, and in JAK1 and in JAK3, so the signal transducer that transduced the signal of IL-15 into the cell. And they did not find any SNPs that were genome-wide significant associated with IL-15 level at the common gamma chain, likely because the common gamma chain is a part of so many different cytokine receptors. So they couldn't pick that up, but they could do modeling studies and basically ask, okay, now that we basically map out the IL-15 signaling pathway, how many patients do we need in order to do that? And one of the interesting findings of the paper is that they need all 54,000 individuals, and is basically the additional value of adding more patients to one of those big populational proteogenomic studies. There is, pretty much a direct proportional relationship between the number of trans-pQTLs that you can identify with the number of patients, whereas that is not the case for the number of cis-pQTLs. So after 5 to 10,000 individuals, you will not find many more cis-pQTLs, but you can find many, many more trans-pQTLs by basically adding more individuals. And this does have implications for pharmacogenomics and for drugs, because cytokines and complement are both common drug targets, especially for antibody, but also for small molecule therapy. So it's just like to bear in mind that JAK inhibitors like tofacitinib and baricitinib are successfully used to treat chronic inflammatory diseases. And then the second part of this study is what Sun and colleagues asked is, okay, what can we do? Or that's one other finding in terms of COVID. Since they did this large proteogenomic study on COVID, they wanted to find protein signatures that basically predict COVID severity. And what they found, interestingly, they found a SNP in the mucin 5B promoter. That's a protein that's released at mucosal surfaces, especially in the lung. That promoter SNP controls 3 other proteins in trans, so that's mesothelin, the lysosomal associated protein for LAMP3, and the surfactant protein D. And that points towards a lung-specific signature that also associates with an increased risk for COVID hospitalization. And that is, of course, interesting because many of these proteins actually have been associated with degenerative lung diseases, like, for example, COPD. So that points towards an overlap of common pathogenesis for severe COVID, but also for degenerative or lung diseases as such. And then they did the same thing for immune signatures, just asking the question, hey, can we find immune genes that in one way or the other have a genome-wide significant SNP that controls protein expression. They did find a gene cluster on chromosome 19 where they had a lot of cis-pQTLs that mapped to different ICAM genes. And they did have a trans-pQTL for the IL-12 receptor beta chain and for TYK2 as well. So that is, of course, also very interesting because it points towards, okay, immune cell extravasation, but also kind of like type 1 interferon, Th1 type signaling that in one way or the other governs COVID hospitalization. So an overshoot of the immune system that basically results in severe COVID. So with that, I wanted to briefly give you the takeaways. So there are three parts that are technical. One of the important aspects of that study, I haven't shown you the data, I'll refer you to the paper, is basically that Olink's Proximity Extension Assay, in more than 80% of the cases, you can actually identify a cis-pQTL for a protein that you assess with a PI assay. That means you can be very, very sure that what you're measuring is actually that protein, because genomic signal and protein coincide. The PI assay performs robustly even in these broad large populational studies, and you do have a high replication rate. I haven't shown you the data, the original data as well. Please have a look at the manuscript, but 96% of the pQTLs that were identified in the discovery cohort were replicated in the validation cohort, despite it being more genetically diverse. And then there is this systems biology part. So these large population of studies, they identify gene protein phenotype triads and can now be expanded to whole pathways. And that, of course, has massive implications for pharmacogenomics. And then lastly, it is the largest proteogenomic study for COVID-19, and Sun and colleagues, they do identify both a lung and an immune protein signature that drives the risk for severe disease. So the next part of my talk is we at Olink, we also have a consortium that is related to protein genomics. That is the SCALLOP consortium. So that's basically for 33 PIs and samples from 43 cohorts, more than 65,000 patients and controlled, that have been measured with Olink's assays, both with targeted assays, but also with explore assays for very, very different complex phenotypes. The SCALLOP consortium is headed by Anders Mellerstieg, and that is basically actively sharing data. So if you wanted to be part of the SCALLOP consortium, because currently our tilt is for European and North American cohorts, you can actually have broad phenotypic data already done. One of the key publications I would like to point you towards is paper Nature Metabolism, where Lasse Folkesen and colleagues actually looked at 90 proteins that were related to cardiovascular disease. So single target 96 assay in 31,000 individuals. And they found 485 pQTLs and could, with Mendelian randomization, infer 18 or something like that, more than a dozen causal relationships between a genomic signal, a protein signal, and a complex phenotype. So with that, I would like to move to the second step, to preclinical research. Where have we been used here? So the paper I found is basically a cell atlas for glioblastoma. It comes from the Stefan Nelander's group in Uppsala. What has been done, or what authors did here, is they had patient-derived cell lines from glioblastoma patients. They more than 100. They did a drug screen with more than 1,500 drugs, and they had this multi-omic approach. So they basically had genomic data available that they themselves derived or via the TCGA. They had transcriptomic data associated with it. They layered proteomic data on top. And they asked questions like, okay, can we find active drug classes? Can we make response predictions? And can we find meaningful combinations that will enable us in one way or the other to overcome resistance to certain therapies? And to make a long story short, the interesting finding of that paper is basically they find a differential bifurcating response for proteasome inhibitors. So some of their primary cell lines are proteasome inhibitor resistant, some are sensitive. And then when looking at the genomic data, what they basically found is that coincided with mutually exclusive aberrations in P53 or the cyclin 2A to B locus. They then took cell lines with that model and basically said, okay, how would these cell lines, based on their genomics and their transcriptomics data, behave? They used Olink's Proteomics assay to add an additional layer, test these cell lines before they put them into xenotransplant models into mice, treated the mice with the respective drug target, with the respective drugs, and then basically found that they exactly behaved like their model would have predicted. And they also found, or they indicated certain drug classes that could render proteasome, bortezomib resistant cells sensitive again. So key takeaway for this paper is that the multi-omics approach that includes proteomics and this pathway analysis actually holds significant promise for the improvement of drug response. And you can even make prediction for drug responses in glioblastoma in these primary cell line models, and that offers opportunities for drug repurposing and fundamentally, hopefully, down the line, the improvement of the survival for glioblastoma patients. Now, I also added 3 studies for clinical development. So what has been done in terms of like clinical trials retrospectively, mostly. So the first study I would like to present here is a study from Antoine Italiano. He's now at the Institut Gustave Roussy in Paris. And that paper was published last year. What it did, it identified the leukemia inhibitory factor as a novel biomarker for immune checkpoint blockade resistance. So immune checkpoint inhibitors are novel prize molecules like PD-1, PD-L1 axis, or CTLA-4 that basically improve the health outcomes for solid cancer patients. So the rendering the immune system more aggressive and basically attacking the tumor. However, there is a subset of patients that does not benefit from these treatments, and that's really where this study from Antoine Italiano came into play. What they did is they took the METCH-R cohort and the premise cohort. These were solid cancer cohorts incorporating sarcomas, non-small cell lung cancers and a small percentage of prostate cancers. The patients had plasma samples taken prior to ever receiving anti-PD-L1 immunotherapy, so that's your Tecentriq, Libtayo, or Bavencios, and they were analyzed with Olink's Explore 1.5K platform or on that platform. They also had the biopsies, so they used the biopsy to do transcriptomics and immunohistochemistry. So they actually did a fair amount of multi-omics as well. But what they found is, interestingly, it's the high levels of circulating lymph prior to ever being treated with immunotherapy actually at baseline already predicts progression-free survival and also the holy grail of oncology, overall survival. So that's really why this paper is so important. And key takeaways is, of course, I mean, it's not only that you can use Olink to really find biomarkers for prediction of immunotherapies, which is a gargantuanly huge market, but you also can identify new therapeutic targets that should kind of like be used in maybe combinatorial approaches together with PD-1 or PD-L1 inhibitors. So that's kind of like the major takeaways from that paper. And then we do have 2 more studies, more into like early diagnosis of cancer. Here, one is basically identifying the captodomain-containing protein as an early marker for lung cancer. Lung cancer is one of the biggest killers in the world. It is the incidence of lung cancer is still on the rise despite prevalence for smoking going down. So Stephanie Danino, they undertook a study to basically find prospective biomarkers. It's a European study, so they took the BioNano and the EPIC cohort, roughly 650 patients and controls. Their validation samples came from the EPIC cohort. It's a independently accrued European cohort. They had medical outcome and they had followed these patients over several years. They had plasma samples, and their hypothesis was relatively simple. They didn't do a broad screen. They basically said, all right, in lung cancer, inflammation has been implicated. So what can we find if we use Olink's Target 96 inflammation panel? And since they had PBMCs for, I think, something like 50 patients and controls, they also combined this with a transcriptomics analysis. And again, just giving you the key findings of this paper. Originally, they identified 12 biomarkers. After the validation cohort, they found that only CDCP1 was associated with risk of future lung cancer development, even after correcting for multiple testing. They also, that was still significant when they corrected for smoking history and for gender, and they had a reasonably good AUC of 0.75 for a single marker. And then when they analyzed transcripts in PBMC, so kind of like a standard, like genomic alterations in these patients, they found a dysregulation of transcripts in the Wnt beta-catenin signaling pathway, which linked kind of like the captodomain-containing protein to early tumorigenesis. So, and since this paper was published in Cancer Research, they actually said that this has potential implications for clinical screening, because together with low dose CT, one should at least improve the surveillance for people at risk. So, for example, people that have an extensive history of smoking, they can use this additional marker for liquid biopsy, together with more expensive low-dose CT screens in order to see if they may develop lung cancer, and then hopefully can be diagnosed earlier. The other study is something a little bit different flavor in clinical cancer research. It's pancreatic cancer. Pancreatic cancer is extremely deadly. It's one of the deadliest cancers, one of the cancers with the worst prognosis. And what is surprising is it's genetically relatively uniform, because it's well known that 95 to 98% of all pancreatic ductal adenocarcinomas actually are in one way or the other have mutations in KRAS, which unfortunately doesn't help to improve diagnosis. So Lindgaard et al., that was their starting point for why would we use proteomics to identify protein biomarkers for early pancreatic cancer diagnosis. And their study design is that who also had a discovery and a validation cohort. They had 700 pancreatic cancer patients. They had they had plasma samples. They had pancreatitis patients, so non-malignant chronic inflammation of the pancreas, and they did have 180 healthy controls. What their hypothesis was also not 100% hypothesis free. They basically said, okay, it is known that pancreatic cancer is extremely immunologically cold, so it doesn't respond to immunotherapy at all. So what we really should test is all the negative immune checkpoints that are the soluble negative immune checkpoints that are included in Olink's Target 96 IO panel. And they combined that with the carbohydrate antigen 19/9 ELISA. So that's one of the very, very few biomarkers in circulation that has relatively low predictive value but associates with pancreatic cancer. And then they had two independent analyses in terms of they had two bioinformaticians going head to head with their respective algorithm to see what kind of biomarker signatures they would find. And what they found is they found two biomarker signatures, 19 proteins, 123 proteins, both in order to have maximum diagnostic value. They had to incorporate this CA19/9 G protein, and they found very, very good AUCs higher than 0.9, and that again illustrates the fact that Olink, even in the targeted platform, is an excellent tool to identify biomarkers for clinical screening of potentially high-risk patients. And this has even more clinical implications, because earlier diagnosis of pancreatic cancer is earlier qualification for surgery, which is hopefully will improve health outcomes, because early surgery or early qualification for surgery is really the only thing that helps pancreatic cancer patients. Otherwise, on the therapy side, there isn't really a lot that currently can be done, which may change with KRAS, with the advent of KRAS targeted therapies. So, and then lastly, I would like to show you a study that's really going more into kind of like the clinical application of Olink's PIA assay, and that has been done together with one of our commercial partners, so that's Octave Biosciences. And what they were interested in is developing a test for multiple sclerosis or chronic inflammatory disease. The issue of multiple sclerosis is a relatively common chronic inflammatory disease. It affects 2.8 million patients worldwide. It's a an autoimmune disease. So basically your own immune system attacks and destroys the myelin sheath insulation of your neurons, of your motor neurons, and that will lead to a tremor and then later to progressive disability. It's a very heterogeneous and complex disease. And even though there are more than a dozen treatments, it's very, very hard to have a good monitoring tool for disease activity, disease progression, or in one way or the other treatment response. So that's really the base question for this study. And then what Octave wanted is basically, do they find biomarkers that point towards relapses, so that potentially more aggressive treatment regimen would prevent those types of flares. So their study design for this lab developed test was basically initially when they partnered up with us, they gave us 82 samples, and they wanted to have a feel for the data. So they did a targeted approach. They took 3 target 96 panels and assessed 220 proteins. And once they were sufficiently convinced that they could read out proteins that they kind of had a hunch about, they actually extended that to the discovery phase. So that was 300 individuals where they did the Explore 1.5K discovery assay, and then they had the independently collected validation cohort that combined samples from 4 different subcohorts, where they basically cut down their biomarker signature to 21 proteins. And then lastly, they came to us and said, hey, could you put that together, those 21 proteins to basically build us our disease activity test as an LDT on the FlexPlex/Focus platform. So that's pretty much the study layout, and here are the 18 proteins that they implicated. They had proteins for immunomodulation, for neuroinflammation, for myelin biology, and for neural or neuroaxonal integrity. And this 18 protein biomarker assay far and wide outperformed anything that is in the clinic right now for disease activity. So that's neurofilament light chain assay, and already has been, replicated in 4 independent cohorts, and is now currently replicated for several thousands of multiple sclerosis patients to basically then be implemented as an LDT and hopefully guide therapeutic decisions and improve MS outcomes. And in case this is a poster presentation, the data has just been released on medRxiv actually yesterday, so 2 days ago for the APAC region. So you can actually have a look at that as well. And then with that, we are at the end of my presentation. I hopefully have convinced you by now that the proximity extension assay is an excellent technique to make new discoveries and develop new ideas about health and disease. And with that, I would like to thank you for your attention, and I'm happy to take any questions that may arise. And here in the appendix, you have all the original literature in case you are interested and would like to have a look at the original data.
Great. Thank you, Michael, for the deep dive into the Olink and how the assays have been used in different stages of research and clinical trials. We do have some questions, and so we'll move into the Q&A. There are a couple of questions related to the Olink Explore, and I'll just ask some of them together. So how is Olink planning to expand the 3K or the Olink Explore protein library? And are you guys thinking of developing it for other species?
Okay, so I mean, what people are very okay. Let's like in terms of the extension. So the extension for the 4.5K, that's basically in the bag. That's going to be released by the end of the year. The exact release date for the product isn't. I'm not like aware, but like the day. But there will be more proteins. So in terms of the library, and we strive towards adding another 1,500 proteins next year. So in case you don't find your favorite proteins included in the next extension and the next iteration of the assay, please just write to us, give us a couple of publications. We will evaluate potential targets, and we always add potential targets to new iterations of the library. And then towards the other question that I guess points towards mouse proteins. So that is a question we get very often. And at present stage, we do have only a single specific mouse protein assay. So that's unfortunate, and we do not have the bandwidth to really aggressively make mouse-specific antibodies to extend it. So yes, it's definitely on the plate. It's definitely something we're thinking about. But the issue is extending the library for needing or making 1,500 new antibody pairs and then validating all of those as kind of like we're slowly but surely reaching the capacity of what a small, still relatively small biotech company like Olink actually can do. So we would love to extend towards mice, but it is a bit on the back burner right now.
Okay. So for interest of time, I'll just ask one last question. Can one translate the Olink Explore findings into the clinic as a laboratory developed test?
Right now, I mean, I wouldn't recommend that. It's simply a cost issue. That is specifically for that. We have basically developed the focus panel. So that's kind of like this translation to make this more economical. And then yes, it's capped at somewhat and 21 to 23 proteins, but that's kind of like why we cap that, because if you want to validate it for an LDT and you want to measure 10,000 individuals, Olink Explore just wouldn't be economical. I mean, even for a large pharma company. So that's kind of like translation wise. Yes, we do work on that. You don't have to use Explore, and I wouldn't recommend Explore simply because it's not economical. If you're just interested in a smaller signature, you wouldn't do that.
Okay, that's good. All right, thank you to those who have submitted questions. That was a great discussion. So if we didn't get to your question, we will make sure we'll follow up with you directly by email shortly. This webinar will be made available as an on-demand video at a later date. So please keep a lookout for the email. And once again, we'd like to thank Dr. Förster for his presentation and all the attendees for joining us today. Right, thank you.
Thanks so much and have a beautiful day.